and welcome to Let's Meet the Virologist, a podcast about the people behind today's virology headlines, people working to understand viruses and how they affect you. We are talking with virologists that belong to the American Society for Virology so that you can learn who they are and what they do. I am Larissa Thackeray, and I'm hosting this podcast from America's Heartland in St. Louis, Missouri. On September 2nd, 2021, we talked with Dr. Adithya Sridhar, a senior scientist in the Organoid labs at University of Amsterdam, who is using gut, airway, and brain organoid model systems to study picornavirus infection. Adithya has a master's in biomedical engineering from University of Oxford and a PhD from the University of Twente, focusing on 3D tissue culture models. Nice to talk with you today. Why don't you tell us a little bit about yourself?
Yeah, sure. So my name is Adithya Sridhar. You can call me Adi. I'm originally from India, from the south of India in Chennai. So that's where I studied. I did my bachelor's there. And after my bachelor's, I moved to the UK and I did my master's in the University of Oxford. So my bachelor's and my master's were in biomedical engineering, so more towards the engineering side, looking at all the different equipment used in the hospitals and so on. And after my master's, I really enjoyed the research project I worked on. And as a result of that, I decided I'm going to do a PhD. I was looking for a lot of PhD positions and I found one in the Netherlands. And in the Netherlands, you're paid as a PhD, you're an employee, you're not a student, you're not on a stipend. So I chose to move here. And then I did my PhD in something completely unrelated to virology. So it was a lot of electrochemistry, a bit of lab on a chip. And my PhD was in the University of Twente, close to the German border. And after my PhD, I wasn't sure what I wanted to do. So I worked in a company as a consultant for a year. And then after that, 4 years ago, a bit over 4 years ago, I moved to Amsterdam. And I've been working in a virology group. And that's what I do now.
How did you first become interested in sort of the science engineering side of things? Were there people in your family that did sort of science, or was it through school? How did you first become interested in it?
Yeah, I think early on it was, I can't really pinpoint an interest because I think it's just one of those things that just happens in India. You know, if you want to study, you do either engineering or medicine. And medicine required a lot more effort than I was willing to put into. So engineering was thrust upon me, let's say. I think one of my family members living in the US suggested biomedical engineering has good scope in terms of finding jobs. So I said, yeah, I would give it a go. And I just did it because that's what everybody did. But I think during my master's, I really enjoyed my research project. And it was kind of funny because there was something very simple. You know, I was doing some cell staining and I was trying to figure out why my stainings were not working. And I spent, days and weeks trying to figure that out. And in the end, it turned out it was something as simple as not having the right filter on the right microscope. And that kind of troubleshooting aspect really got me interested in science. And I said, okay, I really enjoy doing all this troubleshooting. So I would like to do a PhD, and I think that's really when my interest in science really peaked. My time in the UK, and I think a nice thing about studying in Oxford was that they also did a lot of public communication activities. So I used to attend talks from other fields. You could go for a talk in quantum physics from the physics department. They would put it in layman's terms, and I could attend talks from Richard Dawkins and so on. And listening to all these really got me much more interested. And I think that my year there really shaped me towards science.
Right. And so you kind of talked a little bit about how you've had sort of a bit of a winding path. Can you kind of reflect a little bit? So you've done sort of some academics and then you were doing some consulting work, at a company, things like that. What are the different environments like? And what do you like about where you are now?
Yeah, I think the environments have been quite different, even between the academic labs. Each lab has its own culture. And I think the culture that we have in our lab is really what I like a lot. We're very free to work on what we want to do. We have a very nurturing group. So I really like that aspect. And of course, I've been learning about viruses and I love studying about them. So that also helps in terms of the motivation. In terms of working for a company, it was completely different. You're more client oriented, especially as a consultant. You're operating on very strict deadlines. Sometimes weeks to months. So yeah, that's kind of different from academics, where your deadlines tend to be much longer. You're operating in years most of the time, especially if you're a PhD student.
Right.
Yeah, so that was different, and yeah, I think just the fact that you have a client to face to, and and a lot of the times they're paying money, so they don't care. They'll sometimes call you on the weekend, and you just have to respond. Those kind of aspects were, yeah, it was different. But I also really enjoyed my time as a consultant, because I think that it was still not very far off from academics, because it was writing business plans for small and medium enterprises. It was also a little bit of grant writing. So all those experiences that I had there is really useful now. It was definitely a different experience working in a company, and I would definitely recommend people to try their feet outside of academics before they decide where they want to go.
When you were going to these different places, people often wonder kind of what you should be looking for. So what is a good lab? What is a good mentor? What's a good position? So what do you look for sort of in a position?
Yeah, I think that's a very difficult question, because of course you normally have a half an hour to 45-minute interview, and you don't get a whole picture of what the lab is going to be like. I think one of the things that for myself, but also for the people that come for an interview, I always make sure that the people that come for an interview here have time with our PhDs and our students to sit down and talk in private. I think that's very important if you're talking in front of the supervisors, then you get a different picture. And of course, when we want to recruit someone, we sell our group as the best there is. So we always make sure that there's an opportunity for people to go and talk to some of the PhDs that they've been working here for 2 or 3 years, to really get a picture of what the lab is like, and whether it's an environment that they want to work in. And I think that's very important, just to talk to the people that are working there, but also make sure you talk privately.
And so can you tell us a little bit then about, the virus that you're working on and sort of the types of experiments that you're doing?
Yeah, so I think, so the viruses that we study, we primarily study picornaviruses.
Okay.
So we study enterovirus 71, 68, picornaviruses. So what we are interested in is the entry sites. So we're interested in the airway and the gut, which are assumed to be the primary entry sites. And from there on, we're interested in how they get to the secondary site of infection, which is the brain. Then how do they interact with the immune cells along the way? But we also do a little bit of work, so primarily focused on picornaviruses, but we do a little bit of work on HIV. So I think we have 1, maybe 2 projects on HIV, 1 project on SARS coronavirus, of course, a couple of projects on CMV and RSV, but our primary focus is on picornaviruses. My main role when I joined was to set up human models for studying these viruses. Because for a lot of the picornaviruses, you don't really have an animal model. So for example, for picornavirus, we don't really know what the receptor is. So when we don't know the receptor, we can't make a mice model to study it. So we started looking into these organoid models. So stem cell derived organoids, so either from adult stem cells or induced pluripotent stem cells. And what I've done over the last 4 years is set up these models. So we have an organoid model system for the airway. We have a model system for the gut and for the brain. So we have all the different organs covered. And in some cases, we can also make them paired. So we can make it paired airway, paired gut. So it also allows us to study in the same donor how it behaves in different organ systems. So that's really what we do. We try to use these organoid models for studying these viruses. And so it kind of fits nicely with my background, because my background has been a lot in vitro modeling. So during my master's, I worked in a tissue engineering group. And then during my PhD, I worked in a lab on a chip group. So we did a lot of organ on a chip work there. So I set up a 3D culture model for studying cancer, and also developing tools for assessing these models. So that kind of flows into here. So if you think about it, these organoid models are not really made for virology. They are models that are built for developmental biology. And so we need to make modifications to make them suitable for virology. So my engineering background in that sense really helps. So one of the things when I joined this group was that they said, we don't need another virologist. We have enough virologists. We need someone from a different perspective, which I really liked. And that really helps, because we have virologists and clinicians in our group. and then my engineering background, and we put all of this expertise together to build an optimum model system, let's say, for virology. And my engineering background still stays because we continue building these models, but of course I've also focused my attention more towards studying these viruses. But we are also making an organoid chip system where we make a gut-brain axis. So we have a recent EU project called Gut Vibrations, where we try to make a complex gut-brain axis model where we have the gut mucosal model system with the gut-associated immune cells. Then we have a lymphatic or vascular system, and then we have a blood-brain barrier, and then a brain organoid. So, then I also continue doing what I did, but then with the focus on virology.
Right. Yeah, very interesting. What do you say to people that, when you talk about your organoid systems and they say, well, how relevant is this really to virus infection in a person? What can you actually learn from, in a way, an organoid system that you can't necessarily learn from, say, a mouse model or by basically taking samples from people? So what are the advantages to the system in a way?
Yeah, so I think there's two things here. One is that people often forget how these model systems work, right? So there are different types of models, and most of the models that we work on are called discrimination models. So they're very good at modeling a very particular aspect. And then you have high fidelity models that then reflect the whole picture of the whole organism. And you can never have a high fidelity model of a human unless you have a patient in the clinic, right? So what we work with is discrimination models, and I think that's something that we also forget when it comes to the animal models, is that we extrapolate what we see in these animal models to the human situation, but we have to realize that they're discrimination models, and that's one thing to keep in mind. Another thing to keep in mind is that the model system that we work with, the reason we work with these organoids is that they reflect a human phenotype because they're patient derived, and what they can also then do is we can have donor to donor differences, whereas a lot of animal models tend to be clonal and the same genetic background, which is not the case here, which also implies that the costs go up. So those kind of things are not, of course, possible in animal models to really have the human phenotype and to have this donor to donor variability. I think age is still a little bit far off, but hopefully in the future we can also model that, because again, for a lot of these viruses, age is important. I think one of the questions that we always get in terms of these model systems is that people always go, but this is missing or that is missing. Immediately people go, if there's no immune system. But one thing to realize is that we are catching up on several billion years of evolution. We can't recapitulate nature in a few years in the lab. And these are things that we're building on. And, perhaps 50, 100 years from now, we will have a human in a lab system to study these viruses. But for now, these models are discrimination models, and we have to, see them in that light. So they're really good at mimicking certain aspects, and we have to use it for that. I think one example is for polio, for example. It's so well studied. But when using these organoid models, they were able to find a new open reading frame and also show that this open reading frame is necessary for infection. So those kind of things you can start answering when you have a human phenotype in your model system.
Right. Yeah. I mean, we always counter with, it's a way to generate hypotheses that you might not essentially be able to even come across or even think about just using human samples, right? I mean, you can go back to the human patients and see if the hypothesis is true, but a lot of times if you don't have the model system, whatever it is, you don't actually generate the hypothesis at all.
Yeah, absolutely. And I think sometimes you also have to realize that you don't want a perfect model, right? If you think about polio, for example, polio is interesting because of the acute flaccid paralysis, but it causes that in 0.1% to 1% of the case. So if you have a perfect model that can only mimic that 0.1 to 1% of the case, then the amount of money you need to put into before you can do anything is going to be monumental.
Right.
So it's important to see in under what context. And I think it's also important when we go and present these results in these conferences, we have to make sure that we describe what the limits are of our system. And I think that's times people don't do that. And that's where all these criticisms come from. I think if you put it up front that, we understand what the limits of our model are, and we're only using it for answering these very particular questions, then it's perfectly valid.
So can you describe sort of like when you build these organoid systems, what are the kind of experiments that you do with viruses? So, you infect them and, what do you do?
Yeah, so for us, there are several things that we're interested in. So first thing is we're interested in is entry. So when we look at the primary replication side, what we're interested in is we're interested in looking at how does the virus enter. So I'm not talking purely about the receptor, but even one step further, or one step earlier. So which side? So with these organoid models, they're polarized. So we can make a polarized airway epithelium or the gut epithelium. And what we can look at then is whether the virus enters from the luminal side or the subluminal side. And intuitively, for an airway virus or a gut virus, we'd expect it to enter from the luminal side because that is the first side it encounters. But for a lot of viruses, we see that actually that's not the case. For picornavirus, for instance, we see it enters from the basolateral side. So then it generates an hypothesis. Okay, if the receptors needed is on the basolateral side, how does the virus get there? Then we can say, okay, does it interact with immune cells, for example, antigen presenting cells like dendritic cells that capture the virus, take them across, or in the case of polio, for example, it goes via the M cells into the basolateral side of the gut and then initiates infection. So that's one thing we look at, and obviously we're interested in which receptor is being used. We are interested in looking at the cell tropism. So the nice thing about our organoid systems is that they're not homogeneous, so they're heterogeneous. They recapitulate the cellular phenotypes of the organ in question. So we look at that, which cell types are being infected, and I think that also helps us try to understand the receptor. So it helps us discriminate in terms of the receptors based on the cell type. We look at interested in the immune response. So the innate immune response, so what kind of responses? So for example, when we look at Paracavirus 1 and Paracavirus 3, Paracavirus 3 tends to cause much more severe disease in neonates. And then, when we start studying these models, we also start noticing that same as COVID, which becomes an immune disease, we have hints that Paracavirus 3 might be an immune disease because it seems to cause a much stronger immune response than Paracavirus type 1. So those are the things we're interested in, and of course we're also interested in, like I said, with building these organoid chip systems. How does the virus get from the primary side to the secondary side? So then we start incorporating dendritic cells, for example, see if they capture the virus and then transmit them from the gut to the brain, for instance.
Interesting. And do you do things like single cell RNA-seq? Like, how do you look at the programs that are inside of your organoid?
Yeah, so we don't do single cell RNA-seq. So for instance, for cell tropism, we do simple things like immunostaining. We do RNA-seq when we have very specific questions to answer, because it is expensive to do RNA-seq and single cell, of course. But then on top of that, our organoid cultures themselves are quite expensive in terms of the media, in terms of the consumables that we use for the cultures. For example, in our SARS coronavirus work, we are doing RNA-seq to get a better picture of what happens upon infection. And also when we treat the cultures with specific substances. So in this case, we're looking at breast milk and what is the effect of breast milk on the infection of SARS coronavirus. So there we do RNA-seq. But most of the time we try to look at cytokine panels, and then we do immunostaining, and of course we do PCR, and we culture the viruses. We are a culture lab. We started as a culture lab, so we always do titrations.
So what do you see yourself doing in the future? Are you going to sort of stay at your current job? Are you looking to sort of get your own lab? What are you sort of thinking about?
Yeah, so for me, I would like staying here. So it's going great. In the 4 years, we have done a lot of work together. Both my PIs are quite supportive of my position, so they let me lead some of the work. So I also get some last author publications, which is of course necessary for my career. I love the work that we do and the environment that we have. It's a really nice work environment. So in that sense, I really enjoy it. So I have a contract for another, I think, 3 years. I'm actually in the process of applying for my own grants. So I think within this group, I would like to set up my own research line. So I think beginning of next year, I'll be applying for my own grant to become a PI.
Can you talk a little bit more about, so the US system is obviously not like that in a way, like we tend to have sort of like one PI, one lab more. So can you talk a little bit about the system in the Netherlands and how you have, I guess, like larger groups in a way?
Yeah, so of course it's a bit different. It's also a bit different here because we're an academic hospital, and there are different routes. Of course, both the PIs I work under are also medical specialists. So one is a pediatric infectious disease specialist, another one is a clinical virologist. So that's really their primary role, and then they also do research. So then there's opportunities to be a PI, and as a department of medical microbiology, there are several PIs that work in this department, and it's a huge department. So our primary goal is, of course, in the clinic, but then we also do research, and then so that allows for there to be PIs. And it's not enough if you're a PI in the Netherlands, because if you need to have a PhD, the PhD needs to be promoted, and that can only be done if you're a full professor. So you're always under a professor until you become one yourself. So, and they have a kind of hands-off approach, because we almost hardly interact with the professor that promotes our PhDs, for instance. So that's how the system works. And to become a PI here really is you bring your own money in, and if you bring your own money in, then they're happy to make you a PI. Some universities in the Netherlands also have a tenure track system. But we do a lot of collaboration. So for us, having multiple PIs within a group is not really an issue. We can collaborate within the group, but we also collaborate with groups from other departments here or from other countries. So we have a very collaborative environment. So it doesn't really make a huge amount of difference. It's just on paper, you're a PI, which is of course nice to have.
So I guess in finishing up, can you tell us sort of what it's been like the last year and a half for you to be a virologist? During the pandemic, it's sort of a unusual time to be a virologist or to be a scientist. What has it been like for you?
Yeah, of course, it's been a very interesting time, because the minute people realize you're a virologist, I always tell this, like in the last year, everybody's a virologist, so our jobs are becoming redundant. Everybody seems to know more about viruses than we do. So they always start getting into, most of the time it's well-intentioned. I think people want to know. And of course, when people want to know, I think as a scientist who's funded by public money, it's very important that I communicate back the research that we do and the understanding that we have. So I'm always interested in engaging in a conversation with people, trying to make them understand our perspective. It's been a bit difficult keeping track of the literature, so TWIV has been useful in that sense, to keep track of all the literature, and using that knowledge, then try to communicate what we think is going on, and of course, we have been wrong all through the year, and I think that's important that people realize that scientists are not omniscient, and we make mistakes as well. And so it's been humbling, and it's been an interesting experience just trying to talk to people, just doing public communication, even among friends who you normally wouldn't talk about these topics. Everybody's interested in it. I've always been interested in public science communication, so for me, I see that as a nice challenge. Sometimes it's been frustrating, especially when you deal with people that don't believe in COVID or people that are anti-vax. So that's also a different kind of challenge that you have to deal with, and that's something that I'm also learning. How do you approach it? Not to get frustrated, not to alienate people, but to have a communication rather than an argument.
Right. And I guess how is the pandemic going where you are? Obviously, I'm in Missouri, so we're sort of, I think, hopefully getting over our Delta surge. We were unfortunately one of the first places to surge in the United States lately, but it looks like we may be bending our curve. What's it like there?
Well, here things are opening. I've been opening up the last couple of months. There are still some things that are restricted, like for example, clubs are not open. Festivals are not happening, but sporting events are going on. So there is some activities where you can be a crowd. At work, for instance, we still have to keep a 1.5 meter distance, although disappointingly, we don't have to wear masks. I still make it a point to wear masks when I go to public areas, but that's been, I think, the attitude in the West about masks in general. So mask is not, people are not putting masks on. The vaccine coverage has been decent, so most of the people that we do see in the hospitals tend to be unvaccinated. So the cases are pretty stable now. I think in the winter they expect maybe there will be a peak. We don't know. We don't really know what to expect. We don't know to what extent the vaccines are going to cover. But for now, everything is, life is back to normal. In the last year, of course, it's been quite challenging.
Yeah.
Having to work from home, not meeting people. We were restricted in the number of people we could have at home. For instance, at one point it was down to one person. We even had a curfew after 9 p.m. But those kind of restrictions are gone. So life is more or less back to normal.
And do you still have family back in India? How's it like there?
Yeah, a lot of family back in India. In India, well, their life is mostly, well, my mom was able to get a vaccine quite early on, so she was lucky in that sense. Cases are not as bad as how it was earlier in the year. But of course, it depends on where you are, where they are. It's okay. I still advise them not to, go out in public areas too much. And I think at the moment it's not been a big issue. At the moment it's not a big issue. Of course, in the past it was, when there was a shortage of oxygen and beds and so on. But there's still COVID precautions. Like, for example, if you're in a hospital for other conditions, then you can't really have visitors because of the COVID rules. In some parts of India, it is bad because of the rising cases and also unavailability of vaccines. And I think vaccine coverage is a bit difficult in a big country like that. But I think once we get the vaccines to enough people, it should be okay. But my family is doing okay, so that's a relief. But if you have to travel, then when we come back, we still need to quarantine, and yeah.
So have you been able to go back to visit your family? Do you go back often, or?
I haven't been in the last year. I was actually there in February last year, in February 2020, January, February 2020, and then that's when cases were going up. And I think in February, just before I was about to fly, there were a few cases in Dubai, and I was flying through Dubai, and I thought I might not be able to get back. And I thought, or if I do, I thought Europe might close borders actually. And then I was able to get back, and then they were very slow in taking action. But yeah, I haven't been there since, and I don't think this year I'll be going, because it's still an issue, of course. Even if I go, there's not much I can do outside.
Yeah.
Maybe next year, January, February, if things are a little bit better, then I would definitely like to, go back.
Thanks for talking with us. It was great to hear about your research, and we look forward to hearing more about it in the future.
Yeah, great. Thanks a lot.
This has been Let's Meet the Virologist, a podcast about people who study viruses. This is your host, Larissa Thackeray, and thanks for listening. You can find us on Google, Apple, Amazon Music, Spotify, and other podcast providers, or at lmtv.podbean.com.